Hello everyone, welcome back to the channel. In today's biology lecture, we are going to learn about enzymes, their definition, types, properties, enzyme activity and function. This is very important topic for NEET, CBSE boards and other competitive exams. So let's begin. What are enzymes? Enzymes are the biological catalysts that speed up the biochemical reactions by lowering the activation energy. In simple words, enzyme make chemical reaction happen faster and more efficiently without getting used up. Remember, enzyme only increases the speed of a reaction but they are not considered during the reaction. Means ये सिर्फ reaction को बढ़ाते हैं, वहाँ पे वो use नहीं होते हैं. That is very important point. What is a substrate? A substrate is a substance on which an enzyme acts. The enzyme binds to the substrate, converts it into products and then release the product while the enzyme remains unchanged. जिससे एंजाइम बाइंड करता है उसको हम सबस्ट्रेट कहते हैं. ये एंजाइम बाइंड करता है सबस्ट्रेट से, उसको प्रोडक्ट में कन्वर्ट कर देता है. ड्यूरिंग दिस प्रोसेस एंजाइम्स आर नॉट involved, so they remains unchanged. Types of enzyme: there are two types of enzymes, simple enzymes and conjugated enzymes. Simple enzymes contain only the protein part, they do not require any additional component to function. Conjugated enzymes consist of a protein part and a non-protein part also called a cofactors. These cofactors are of three types: coenzyme, enzyme. Coenzyme is an organic molecule that helps the enzyme function. Example is NAD. Prosthetic group is a non-protein component that is tightly bound to the enzyme. Third is metal ion activator. Some enzymes require metal ions such as zinc, magnesium for proper activity. Protein plus cofactor becomes the active enzyme. Properties of enzymes: enzymes are protein in nature. All enzymes are protein in nature, but all proteins are not enzymes. That is very important. They are highly specific, meaning each enzyme acts only a particular substrate. They work best at optimum temperature and optimum pH. High temperature or extreme pH can denature enzyme, causing them to lose their activity. Optimum conditions are essential for maximum enzyme activity. Now, what is enzyme activity? Enzymes have a special region called the active sites. This is enzyme and this is the active site. The substrate binds to this active sites where the reaction takes place. There are two important models to explain the enzyme action: lock and key model, and second is induced fit model. According to lock and key model, the active site of the enzyme has a fixed shape. Only the correct substrate can fit into the active site, like a key fits into its matching lock. The enzyme form an enzyme substrate complex, convert the substrate into products and finally releases the products. The enzyme remain unchanged and can be used again. Here this is the active site where the substrate fits. Next is induced fit model. According to this model, the enzyme is flexible. When the substrate approaches, it can change their shape slightly to fit the substrate perfectly. This model explains enzyme action more accurately than the lock and key model. By doing this, enzymes catalyze metabolic reactions, provide an alternative reaction pathway, lowers the activation energy. Function of enzymes: enzymes perform many important functions in living organisms. They catalyze metabolic reactions, making them much faster. They help in digestion, respiration, DNA replication, protein synthesis and many other biochemical processes. Without enzymes, most reactions inside the body would be too slow to support the life. Quick revision and points to remember. Enzymes are biological catalysts. They lower activation energy and speed up reactions. Substrate is the molecule on which an enzyme acts. Two types of enzymes: simple enzyme, conjugated enzymes. Cofactors are coenzymes, prosthetic group, metal ion activators. Enzymes work best at optimum temperature and pH. There are two models of enzyme: lock and key model, induced fit model. Enzyme remain unchanged after the reaction. Enzyme works, product formed, enzyme stays unchanged. Remember this: enzyme works, product forms, enzyme stays unchanged. That brings us to end of the today's lecture. I hope this explanation made enzyme easy to understand. If you find this video helpful, please like, share and subscribe to the channel. Don't forget to press the bell icon for more biology lectures. Thank you for watching. See you in the next class.